Whether you keep them in your home or love to see them in theirs, these are the creatures that bring us all together.
Reptiles. We're going to be delving into the experiences of reptile lovers from around the block and around the world. This is the Reptile Talk Podcast. What's going on, everybody? My name is Jeremy Turgeon from Brassman Reptiles.
And I'm Rob, and I'm keeping it real.
All right, man. So this is episode 3.
Boom.
And I'm pretty stoked because it's almost like a kind
throwback.
of a throwback.
Yeah.
Because we're joined by the one and only mad scientist himself, Mr. Steven Tillis. What's going on, Steve?
Thank you guys for having me. Not much, same old same. Lots of lab work, lots of snake work.
The best things, you know.
The best things.
Yep, yeah, that's for sure. Yeah, I guess I'm in the lucky place where my two get to intersect. So
Right?
That is true.
Not everyone gets to do that.
So I want to, for those people who are like just checking this out, we talked a little bit on the very first episode how like we did this podcast 7 years ago?
A while ago.
Yeah, that's kind of a while ago. So before we really dive into some stuff, let's throw it back a little bit. What the hell were you doing 7 years ago other than making me feel like an insignificant piece of shit?
Oh, yeah. So I guess 7 years ago, that was when I just started undergrad, and so I did my undergrad. I did my undergrad and now my PhD at University of Florida. My undergrad was in wildlife ecology and conservation. So at the time, the snake collection was, I don't even remember what I had. It probably not a lot, less than 100 snakes. Yeah, I was just getting in the groove of school, and just started working for Eugene Bessette around that same time too. So yeah, that was a long time ago. That was
a lifetime ago.
Setting the foundation, I guess, for what all I'm at now.
Yeah. Now, and in the midst of all that, your personal collection has changed quite a bit too.
Yeah, it's ebbed and flowed over the years. I think that was when I really first started scaling up on the blood pythons. I think before that it was mostly carpet pythons and ball pythons and stuff like that. And I had some blood pythons, but yeah, got hooked in line with the right people and really started scaling up on the blood pythons.
I remember that because I remember seeing photos and was just like, he's done it again.
Yeah, I can never take really too much credit for any of the stuff I've been lucky enough to do. It's all just been stupid luck in the right place, the right time kind of thing. You know, it's never part of a grander plan or anything like that. So yeah, that was just meeting the right people at the right time.
There's always a grander plan. And speaking of meeting the right people, so you were talking about Eugene Bessette, and when we were just down there at Carpetfest, you had kind of helped us out to go check out his place. So where did you meet Eugene initially?
So I met Eugene for the first time when I was 16 at a reptile expo. I was vending the expo down in Orlando, and I think I had some green tree pythons on the table that I had produced, and he was walking around and saw them. And for those who don't know, Eugene originally, how do you even say originally when you're talking about Eugene?
Right.
At some point during Eugene's career, he was really well known for green tree pythons. And so, you know, he said, oh, those are nice looking green tree pythons. You know, how about you come check out my facility at some point? And then, you know, I think we had a off of school. And so I drove up here to Gainesville. I was living in Orlando at the time. And that was how I first got introduced to Eugene, is seeing his colony. And the colony looks very different then than it does now. And then when I moved up here for undergrad, I ended up buying a property that's like literally, as a crow flies, maybe a quarter mile from his facility. And so, you know, I was like, hey, I'll be looking for a job. I'm moving up there. You happen to be hiring. And yeah, I guess the rest is history.
That's crazy. So when you first started working with him, were you just another shit cleaner?
Yeah, no, for years. For years that was my role. It's basically, you know, just generic cleaner. And then as the more years I spent there, the more I started going to kind of expos with him and stuff like that, and kind of getting a full feel of how his business flows. And then that's what kind of, if it weren't for those foundational years of just years of knowing how his business operates, how the workflow goes, and all that stuff, put me in the position to build the databases that I did that are so ingrained with the workflow there. And so yeah, it really started as, again, another happy accident that I can't take any credit for planning so much as just, because I had done so much grunt work at the facility in the first place, I knew exactly where a database could fit in line and really improve or work in line with workflow.
Yeah, and that database is
impressive.
Something out of
the future.
The future, literally.
And honestly, it's just the beginning in terms of the potential of what we can really do with it. I have some. If you come back in a year or two, it'll be even more impressive than how it looks now. And again, it's not something that I could say, from the beginning, I fully envisioned what it would end up being, but I don't think there's another facility on the planet that is anywhere close to the data system that Eugene has.
Yeah, so Eugene has always been super big on keeping records, and he's kept really thorough records for years and years and years. So when it came to your database, you were working outside of Eugene's place with that database, right? Or did you kind of build it to fit his?
No, I built it exclusively for his place. Yeah, honestly, that database, as it stands now, would be almost outside of that facility because it is so ingrained into the workflow, and that's why it works.
Yeah,
there's a lot of reptile databases available out there, and it always feels like you're trying to conform your collection to the database instead of having this opportunity to build something literally from the ground up and build the database for the collection. Now, that also means that database outside of the context of that collection would be, just as semi-cumbersome to implement in a different facility than any of the other databases, if not even more so, because it is just so customized to the workflow. But at the same point, it puts us in a position where we can take an employee and have them really, drop them into not only the system in terms of how we, clean cages or manage the animals or anything like that, but also to plug them into that database. So it's all one thing. The database and the workflow are the exact same thing, where the two are so married to each other that it improves our efficiency drastically. But at the same token, if you took that, just one of those two components and tried to implement them elsewhere, I don't think they'd be anywhere near as useful. It really is the customization that makes it so powerful.
Yeah, it's absolutely amazing. Having seen just a glimpse of it, I mean, I still stare at that printout that Eugene had you do for me, and I'm like, holy shit! Like,
it's impressive. Like the amount of specificity to it is what really impresses me.
Yeah, it's really handy because I still kind of consult on managing the colony, but basically I'm there for maybe 10 minutes a week. But because of this database, I have this bird's eye view. Basically, I feel like my finger's always on the pulse of the colony, even though I'm not there at all.
Yeah.
And that's just because of the type of data and the longevity of the data and the breadth of the data that we've been collecting.
Yeah, and one of the things that I thought was really interesting about it was so Eugene's been keeping records for decades. And when you guys started using that system, you only input, I think you said 3 or 4 years previously, and you were already able to see really specific trends in that, which I think is amazing that in even just a short period of time, you could see so much.
Yeah, with the collection size that he has, it doesn't take very long to get a really large sample size and start looking at some really interesting patterns. And honestly, that's just the beginning of it. Now, where we have it now that we are, so this was our first year implementing some of the barcode stuff. Now the next goal is to scale that out to a whole different level where basically what I told Eugene is that 3 years from now, the system will be picking out our holdbacks based off of the feeding records of the parents.
Mm-hmm.
Oof.
That's pretty cool.
Sheesh. That's I can't even. That doesn't even register for me. Holy crap.
Yeah, it's a power that I don't think. Again, it's one of those things where the more layers you add, the more you can kind of see what you can do with it, and yeah, I think the potential is through the roof in terms of what the data can tell you.
Man,
and
that's crazy.
So the biggest thing too is that, and the hardest thing too is that it's one thing to just collect data, and so for Gene for the longest time had paper cards for all of his animals, and so it was all just paper data collection, and that's really good data to have, but it's not. You can't just snap your fingers and have that magically jump onto a spreadsheet in front of you.
Yeah,
so
right.
There's collecting data for data's sake, and then there's figuring out how to collect data in a way that you can actually mine patterns and pick out particular trends and stuff. Basically, that data needs to be easily mineable. It needs to be easily accessible,
right.
and that has been the biggest change, I guess. And it was lucky in that Eugene is so data oriented that basically it was like, this is the same thing you've been doing, except for now you have a magic wand that tells you everything without having to go and look at 1,000 data cards.
Yeah, that's magical. It really is, because I remember coming back from that trip down there, like that was probably one of the top 5 things that was like stuck in my brain was like the intensity of that system, and then like thinking of a way to like bring that back in some shape to what we're doing at Nerd. And I still have not figured that out. But being able to like input some data, just throwing stuff on a spreadsheet for what we were seeing with just last year's pairings, like I was immediately able to see certain trends or like where things were in racks, how that affected like the temperature difference from one side of the room to the other, and what we were seeing in correlation to clutch sizes and how well some of those females did. I was like, oh, that's actually pretty interesting. And that's literally just throwing something in Microsoft Excel and just being like, yep, there it is. And then being horrified at the amount of ball pythons that are in the entire building.
and we're saying that, and we are producing 1/6 the amount of ball pythons that Eugene's doing.
Yeah,
so.
exactly. Yeah, let's not talk about those numbers.
It's impressive. It really is. And I say that very humbly because, again, it's not like I'm like, oh, I have a brilliant idea of how I could build a giant reptile database. It's like, okay, I'm tired of dealing with this Word document of all these printed IDs. This is not a good way to do it. All right, let's just have a spreadsheet. Okay, let's turn that into 2 spreadsheets that talk to each other, which is all a database is. And then you just, the product that you built, that you saw took me 5 years to get to that point of tinkering with it and saying, okay, well, this worked. Now let's add one more layer of complexity, and so on and so on and so on. And it's not like, again, it's not like I had an original vision for any of it. It just, I stumble my way through it and realize the power that I've accidentally built into my fingertips,
Jeez. Dude, you're an evil genius.
Yeah, that's my business model now at this point, honestly. Yeah, I still have really what I've shifted my colony towards is a wide variety of python species, but low numbers of a high diversity of pythons. And what I've turned my business into is basically I tell myself I don't ever want to sell another snake to a customer again. Like I'm so far over it that a word or a sentence you'll not hear in the future is, oh, I got that snake from Stephen Tillis. Like, it's just, I don't want to deal with I'm over it. So now I've turned my facility into what I call it is the experimental laboratory of herpetoculture. I've turned my collection into, there's just mad scientist tinkering stuff. And, with any of that, the failure rate is always incredibly high whenever you're trying new different things. But at the same time, you occasionally stumble into a good idea and it does some cool things.
Yeah, for sure. For sure. So another interesting point is you're probably the only person in probably the world that has actual locality ball pythons.
Yes, yeah. Yeah, so so I've done 2 trips to West Africa,
Mm-hmm.
and I was actually there for pangolin conservation stuff.
Yeah, just rub it in. Go ahead. That's fine.
Yeah. It was, again, a happy accident that I get to, stumble into a project that happened to be taking me to a part of the world that I already had interest in.
Mm-hmm.
And so while we're there, because we're, dealing with governmental conservation entities anyway, we were kind of plugged into that wildlife network anyway. So I got to, tour all the farms and stuff like that, which was really cool and really got an understanding for how these farms work. And so, yeah, how these farms work is when a villager stumbling around West Africa finds a gravid ball python in a rat burrow in his cornfield or whatever, they'll collect that gravid female. They send them to a central location, usually in the capital of whatever country. So there's a lot of those businesses in Accra in Ghana, and then a lot in Lomé in Togo.
Mm-hmm.
And so there the females lay their eggs, and then the babies are sold if they can get a good money for them. If not, they'll end up actually releasing a fair bit back into the wild. And then after the females are done laying, they get sent back to the wild. At least that's how it has been operating. Now that so there's a little side tangent. We're producing normal ball pythons for so cheap now that the export CITES is actually about the same price as the snakes actually sell for. Mm-hmm. And so now those snakes are not worth anywhere near as much as they used to be. And so now a lot of those females, instead of going back into the wild to then collect next year, are going into the skin trade. So it's again, a little side tangent that our production of ball pythons here has direct relevance towards their captive counterparts, because what before was kind of it seemed to be pretty sustainable, honestly, of ranching ball pythons and then releasing females and some of the offspring back in the wild is now just, collect, sell the offspring if we can, sell the parents for skin trade. So when those females are redistributed out into the wild, a lot of times the information from where they came from is not really paid attention to. It's, you brought in 300 ball pythons, and so here's 300 ball pythons to put back out.
Mm-hmm.
And so because of that, across their range in West Africa, I wouldn't put really any credence whatsoever into a true locality ball python, because it's they're being kind of brought to a centralized location, playing, musical chairs, and then tossed back out into the wild. So what we had done with part of this project is it took us to middle of nowhere West Africa, in Togo, and so the whole region we were in is in the Volta region, and so you see a lot of ball pythons tagged with the name Volta, and it has absolutely nothing to it. Volta is a locality that's like half of the ball python range is technically in in the Volta region, so it's like a nothing name to go along with it.
That's perfect.
It's like getting a corn snake from the eastern United States. You're like, yeah.
Exactly. But really all these Volta females are just really old ancient ball pythons that happen to live long enough without being eaten by something or someone. But you slap a cool tag on it, and now it's a Volta, and you could ask for a lot of money for it.
Right.
But anyway, so we were in the Volta region, and we got to go into some like really middle of nowhere type places. And there was this one French guy who lived in one of the villages who was like the one guy that whenever they found a snake that they were scared of, he would come and remove it. Mm-hmm. And on his front porch, he had this wire cage with like chicken wire cage with like a python that he had collected from the local area. in it. And so in that cage was this 6-foot, 2-inch female ball python, and then I picked out a male to go with her, where basically I know the exact locality that these ball pythons came from to the village. And it's a village where the trade isn't developed in any kind of way where those females will be swapped around. So it's basically a true locality ball python that is, I'm vehemently opposed to attaching the name Volta to it, but from the Volta region, this giant female. Now, that being said, she was a giant female that we found. Every other ball python that we found was far well within the range of a normal ball python size. So, which is why, again, I said that the locality thing is kind of a bull.
Yeah.
But anyway, so yeah, I have these locality ball pythons that I brought back from West Africa, where we went to that village, and I ended up trading the guy a pair of snake tongs for it because he had just gotten bit by a stiletto snake like a month before because he was using like a piece of bent rebar or something. Yeah.
Oof.
Ouch. That's
actually, the stiletto snakes were the most common snakes that we found there.
Really?
Yeah, it was actually the first snake we found, because the first one I found was like 6 foot up a tree, and we almost grabbed it because I'm like, there's no venomous snake that looks like this that would be living in a tree.
Right.
Mm-hmm.
And then right as I go to grab it, I'm like, that looks awfully stiletto-y. Let me hook it. And then, the second I hook it, that fang comes flipping right out and jabs that hook. So I'm like, well, that would have been a short trip. So,
yeah, that would have ended the whole trip right there.
Damn.
Yeah. So yes. So anyway, I traded him a pair of snake tongs for this pair of ball pythons and brought them back, and was finally able to get them to breed. And so these are probably the only true locality ball pythons that aren't just tagged with some bullshitty name, that aren't mixed from a bunch of different localities from the farming process. So it's it is something very unique, and it's something that, again, I was actually impressed by how much people liked them, because at the time, I was like, me and 4 people are going to care about this, and everyone else is going to be like, why'd you waste the postage?
Yeah. So now what you're going to be able to do is you create a whole vein of locality-specific ball python lovers that hate everybody else.
Yeah.
Because that's pretty much what exists in every other aspect of the industry. So
yeah, the only thing the ball pythons are missing is the pretentious locality people.
That's
Yes.
true. So now you can start that vein of people. And we'll look back and laugh and then call you an asshole for it.
Yeah. I mean, that female is a really big ball python.
It really is. No, it definitely
It's a
really
really
is.
big ball python. And there's a difference, and this is why I say that they're just really old females, is because it's not just that she's a big ball python. Like, you can raise a big ball python if you feed the ever-loving snot out of it.
Yeah.
That's not what this snake is. This snake, the head on it is twice the size of any other ball python I've ever seen of a comparable size. And that is something that I cannot duplicate in captivity. So I also have a project of South Florida locality Burmese python.
Mm-hmm.
And if I pluck a 6-foot snake out of the wild and then compare it with a 6-foot captive snake that I've raised, it does not matter what diet I've fed it. It doesn't matter if I've tried to get it big in a year, or if I've tried to, I have some animals that they're like 2017s and they're just 6 foot long because I've fed them so slowly. Does not matter. The head on that wild caught snake is going to be twice the size as any captive burm that I could raise. And I have no idea what the mechanism.
Okay. All right. We're still recording.
We're back?
Yeah, we're back. I don't know what the heck that was.
Sweet.
Yeah. Okay. So it just cut us off when you were talking about. So, the head of that animal will always be twice the size, and you don't know what the mechanism for that is.
Yes. Yeah. So I don't, and that's with the Burmese pythons. But again, I've never seen a captive ball python that's been raised under any feeding regimen that has that same size, but also that same head size. That's natural grown, cannot fake it any other way. We've not perfected the science type growth. that again just makes me think that these are just really old females. They're not anything special genetically, other than that genetically, I guess, live a long time.
That's interesting.
That is really interesting. It makes me wonder if the high competition or just having to go with food items that might not be like, in captivity, we're giving them food items that we think are perfectly sized for them. So something that they can very easily overpower. And then sometimes you see pictures of wild snakes or videos of wild snakes where they're eating these giant things that you're like, one, how did they kill that? And then two, how did they swallow that without tearing something? And it makes me wonder if that any of that plays a role into it, or just more the natural sunlight, or the competition with predators, or what it is that is causing that.
Yeah, so I've heard some researchers, and this is just spitballing. There's no data that goes. I think it might be some sort of ontogenetic based off of just that massive prey size that you're talking about.
Yeah.
Be it a South Florida berm or be it a West African ball python, they're almost certainly at some point going to get a point where their eyes are too big for their stomach and end up eating something that no captor would have ever given them an opportunity. And maybe that in and of itself kind of stretches the jaws that lead to some sort of genetic
growth
change in the gene expression there.
That's really interesting. So kind of like going in line with this. So you went to that part of Africa for pangolin research. You've done a lot of traveling for research projects. I know you I don't know if you still do, but I remember seeing that you were doing some sea turtle research and stuff a few years ago.
Yeah, I've done some sea turtle stuff down in Costa Rica. I've done a project in Grenada doing actually ultrasounding the Grenadian tree boas there, which was really cool.
That's really cool.
That's freaking awesome.
Yeah, so I've gotten, when you're young, and now that I'm markedly less young than I used to be, the magic has no longer there. But when you're young, you can ask people to say, hey, can I partner up on your field project? And usually they say yes. So if you're young and you're looking to get experience, just ask. And that's how that's 3/4 of my secret to being where I'm at now is just ask.
If you don't ask, the answer is automatically no. So
that's true.
Yeah, that
is true.
And then you went recently to Australia too, but that wasn't for research. That was for fun, right?
No, that was for fun actually. So yeah, the Australia trip was actually logistically intense. So we did an Australia trip because I was actually presenting at a conference in New Zealand. So it was a New Zealand Australia trip. So the New Zealand part was actually that was for research or research affiliated. But our goal for the Australia trip is like we want to hit every bucket list Australia item we can, so that in the future when I do other Australia trips, I don't have to feel guilty like I'm missing something when I want to go target red lice python or whatever in the middle of nowhere, where it's like, if I don't find one now, I went to Australia and did nothing,
Yes.
So yeah, we hit all the touristy stuff, and that way my bucket list is satisfied on Australia. So now I can go do the stuff that I really am, wouldn't mind wasting a trip if I fail over,
Yeah.
But you did see some reptiles out there, right?
Yeah, we actually only had a couple of nights of like decent herping, but yeah, we found a carpet python and a pair of spotted pythons and a bunch of keelbacks, bunch of the brown tree snakes.
Cool.
We had a decent luck herping, honestly.
That's awesome.
The carpet python especially. That one was like, there's very, those moments when you're herping and you like spend hours picturing in front of you what something is going to look like to the point where you can almost like convince yourself it's there.
Yeah.
And then it is there, and it's so perfectly exactly how you were picturing it that it just doesn't seem real. That was that carpet python.
That's
awesome.
That's pretty much how it was for copperheads with me. I was like, oh man, it's gonna be sitting in a pile of leaves and its tail and its face, and then I was like, is it actually? Oh my god, it's sitting right there.
Yep. Oh man, that's amazing. That's amazing. I definitely want to take a trip to Australia one day.
Australia
when-
is big up on my list, but
Yeah.
if I go there and I don't see a Bell's Phase lace monitor, I'll cry.
Oof.
Yeah, there was.
Setting the
We
bar
didn't see
high.
any the Bell's phase, but we saw. They're not to the level of being a garbage lizard there, but the lace monitors are just
Fairly
everywhere.
common.
Yeah.
Yeah. Those
So,
and
and
death
honestly, I
adders.
like. Yeah, I was really bummed. We were actually when we were looking for when we found the carpet python, we were actually targeting death adders. We were in a really good death adder spot, but had no luck on that. But it was really hard to complain because it was, carpet python, and there's also echidnas and really cool mammals and quolls and stuff like that. So I'm a biologist first and foremost, so it's my love is not limited to just the snakes, but
Right.
it was a cool trip.
Okay, so now here's the really
then,
important question about the carpet python. What kind of carpet python was it, Steve?
They're all carpet pythons. Come on now.
Yeah, it was a coastal.
Okay, all right. Otherwise, people would be like, well, what species? Bruh, it's important.
I don't know.
Mr. Zio River locality ball python.
Oh my god! And it was a really beautiful carpet python too.
It was. It actually, it was a really nice looking one, and that makes sense given the locality that it was in. It was like close to tropical, but not quite tropical. And if you think of a jungle carpet python as just a jungle python, it makes sense that what you'd have would be kind of similar looking to both.
Yeah, yeah, kind of rides that line.
Yeah, for sure. For sure. Damn.
But yeah,
I
and
wanna
then
go to Australia.
New Zealand was really cool too because they have all the cool geckos and
Naughtiness.
stuff like that. And I got to
Naughtiness.
play with the tuatara, and that was it. I was like, oh, I'm never gonna be cooler than I am right now.
It's hard to beat tuataras, man. They're
Yeah.
just so ancient.
They were way more different than I was expecting. I mean, don't get me wrong, they're clearly different than anything else, and that's one thing to conceptualize, but it's a different thing when you're working with one. They're a different beast.
That's so cool.
Yeah, that's badass.
Did you see any of the geckos out there? Any of the Naughtiness or anything?
None in the wild. They were all in a captive setting, but
Still.
yeah, it was still really cool. We saw the Auckland green geckos. There was a couple of those, and then the Otega banded skinks. Those were really neat. Yeah.
Cool. That's pretty awesome.
That's awesome. Damn it. I just want to do cool things like Steve.
I want to go to Southeast Asia. Southeast Asia is like my dream area to go
Yeah.
to.
Yes.
Southeast Asia is next on my list, but the hardest. I've been struggling because it's such a vast area of how do I do a trip to their justice.
Yeah.
Mm-hmm.
Yeah, there's so many different islands. There's so many different species that you could target while you're out there. And, not every place wants exactly, people like us to go out there. So it's tough to kind of narrow it down to what you would want to do or where you'd even be able to go,
Yeah, most definitely. I definitely am on that train though. Like Southeast Asia is up there. Yeah, there is just so much
I want
fucking
to go to
ground
Borneo
cover.
so bad. Borneo and Sumatra.
Yeah.
Ever since I was a kid.
I never
mean-
would have guessed that.
Never?
Yeah.
No,
Really?
never would have guessed
It's
that.
the clock's ticking on a lot of areas in that part of the world.
I
So
know.
I'm
It's scary.
it is. Yeah.
So what we're saying is group trip.
2021.
Yeah, I need to get some good contacts on the ground, and then that's usually how you can get the guarantee of a best trip is to know someone local, and they can
Mhm.
save you a bunch of waste time.
Yes.
Yeah, for sure.
Yeah. Yeah.
For sure. All right, so I want to know what. So you're doing all of this work. You've got everything that's going on with Eugene, your degrees, what you're doing with your own personal collection. But then there's also, we haven't even talked about this really much at all. Your involvement with the Nidovirus research.
Yeah, I was going to say we've done this whole conversation without even mentioning the word virus, which is extremely pertinent right now.
I know. It's very true. Yeah, so you're one of those guys that's like on the front line of the research for this. And damn, there's a lot of stuff.
There is so much, and going to Southeast Carpet Fest and hearing you and some of the other people on the panel talk about Nidovirus and the research you guys have been doing and the ways that it presents itself and all these different things that fall into line with it or about it. It's like when people hear about Nidovirus in any of the reptile community, it's like an alarm goes off, people are losing their minds, and everybody's freaking out, but then they don't know anything beyond, oh, it's scary, you should be afraid of it. And then listening to all the different research you guys have been doing, it's more in depth than that for a lot of people, or how it can play out.
Mm-hmm.
Yeah, so this is an interesting conversation to have right now. And the reason is, and a lot of people don't realize this, but coronavirus is a nidovirus. Like, it is within the same taxonomic order as all of the snake nidoviruses that we see. It's literally, there's snake nidoviruses, which are serpentoviruses, on one branch of the same tree, and then there's coronaviruses on the other. So a lot of the parallels you're seeing now should sound awfully familiar if you've ever been listening in on the snake nidovirus stuff. The whole asymptomatic carriers, the whole, why we have, in some contexts, it can totally destroy and kill significant portions of the population, and then in other contexts, it just seems to be there.
Mm-hmm.
It's really funny because whenever I give my talks about the snake nidoviruses, I always talk about, why it's important, and I start by talking about SARS, and now it's like my talk introductions have written themselves for the next 10 years.
Yeah, you just point at the next television, you're like, you see that right there? That is it.
Yeah, exactly.
Yeah, exactly. So my research has become incredibly relevant on a very short time frame in terms of even just beyond the snake community. But that is what makes it so complicated to explain to people, is what I've been telling people is that if it has a lung, and honestly, even if it doesn't, there's probably a nidovirus somewhere in that species. Whether or not we found it yet remains to be seen. There's nidoviruses in insects, in cattle, in people, clearly, in all sorts of reptiles, again, you name it, it has a nidovirus. And these nidoviruses are sometimes very, very different from each other. I mean, we see some snake nidoviruses that are, 50% divergent from each other. So
wow.
Very different viruses circulating. And it's really hard to explain to people that, like, virus doesn't necessarily equal death. It's there are some contexts in which some species of viruses in some populations can be a total death sentence, and yet you have other populations that have viruses circulating at a percentage that would make you genuinely squirm, like 30 to 50%, without any disease whatsoever. So, but is that necessarily true if that virus jumps ship into a different species or a different population? And that's the questions that we're trying to answer, is which viruses matter in which contexts? And that's not as straightforward as a puzzle as it's easily digestible for most people,
Right. Yeah, it seemed like when we were chatting with you, and then when we were at Southeast Carpet Fest, hearing more information on it, like there's, I mean, like you guys, as far as like the serpentoviruses are concerned, like it's still very much in the infancy of research. Like there's still a lot of questions to be answered.
Oh yeah. Yeah, there's a ton of questions to be answered, and we have made a lot of legwork so far, just of the studies that have been published, and there's a bunch of stuff right now that projects that I'm working on finishing up that'll be pushed towards publication, and will be hopefully answer kind of bridge some of the gaps of understanding, and honestly, although after I complete my PhD, I expect to end up with more questions than I have answers at the end of it. That's how. Yeah. How complicated some of this stuff can get, but it's there's a lot we just don't know yet, and there's a lot we're continuing to understand. Now, a lot of what we do know, or a lot of what we assume we know, is based off of research on coronaviruses. So, a lot of the broad statements we make currently, we can say, okay, don't know that for sure for serpentoviruses, but the next closest thing we have are coronaviruses, and this is how they act. So
Right.
now what we're doing is the research to say, okay, what about these viruses specifically? And so a lot of the studies we're working on now, like which disinfectants kill it, and which, how efficient, how long can it last in the environment.
Mm-hmm.
So that's one of the first aspects of the studies that we should be pushing out first is how long they can last in the environment. So it falls within close to what we'd expect with some of the other stuff based on coronaviruses, where honestly, in terms of their how stable they are in the environment outside of a week or so, they're not very stable. And that is the benefit of nidoviruses, is that they're enveloped viruses, and so all you have to do is break that envelope, and they're no longer a functional virus. So you're of the mindset that you're battling the virus, but in all actuality, what you're doing is you're battling one single layer of fat, and once you break that, that's it.
Yeah. So if you had to really dumb nidovirus down to somebody who's like, I just heard about this thing, I don't know anything about it, but I'm now like horrified, how would you describe nidovirus to that person in the most like layman's terms ever? Because I know like one of the things that we've seen, and like, I mean, as soon as you get really into like the arboreal snake community, like it seems like for a while there was like a nidovirus post like everywhere, just like trying to get more information about it. And because there's so many unanswered questions, like I get frustrated trying to explain it to people because we just don't know everything. And they're like, well, what about this? And what about this? And what about this? And they just expect there to already be this documentation of it. And a lot of these, like a lot of the newer keepers that are just starting to find out about it, don't understand why there isn't all this research already done. Like we had a customer recently who was like, yeah, I get all my stuff tested because like blah, blah, blah. And, is like, I'm like, well, what does your test result look like? And he's like, it's a piece of paper that says nidovirus negative, and it's like, okay, well, what strain, what species, there's so many extra questions that like we just don't have answers to right now. So if you were going to explain this to somebody who had like zero understanding, what would be the easiest way to kind of break it down to them?
Sure. So, it's a group of viruses, and I guess I'll talk about specifically snake nidoviruses / serpentoviruses. So that they're a group of viruses that target the oral and respiratory tract of a lot of different types of reptiles. They have viruses in snakes, viruses in turtles, viruses in lizards. They're all targeting the oral tissue and can make it into the lungs, and that's where you start to see fatal consequences from some of these viruses. So some of these viruses have the potential to wipe out colonies. They can and they do.
Mm-hmm.
Others of these viruses don't seem to be as medically significant in some contexts. So the easiest way when you're talking about nidovirus is to prevent it from getting into a colony in the first place. And that comes down to testing.
Mm-hmm.
Now, the important part of testing is to know that not all testing is created equal. And it's definitely one of those things where, a $25 test is not the same as a $60 test or a $100 test kind of thing.
Right. So what's the biggest difference between that?
So a lot of your cheaper tests are what's called a qPCR, and it's a different type of PCR test. And so a PCR test is just looking for any bit of, in this case, RNA of the virus, amplifying that. And then, if it's there, it'll either come back as positive or negative, that you were able to replicate RNA, that you were able to find that RNA. Now, a qPCR has a unique ability in that it uses this fluorescence, so it can actually quantify, which is what the Q stands for. It can quantify the amount of virus in the sample. And because of that, it's actually a more, not a more accurate test, but it can pick up virus in lower levels than a standard PCR. Okay. But the reason it can do that is those fluorescent primers need a lot more specific of a sequence. And so a qPCR test, if I know that my ball python colony has ball python nidovirus in it, then I would use a qPCR for ball python nidovirus, because I'm only looking for one type of virus, and I already know what that virus is, and I know that what's in my collection can be detected by that test. So that's why you see some of the cheaper tests are like for ball python nido or for green tree python nido, and not just nidovirus.
Right.
For that is because they require far more specific sequence that will miss divergent strains if you don't already know what you're looking for. Now, the more expensive tests, and the reason they're more expensive is they're a lot more labor intensive, is your standard PCR test. And so that is going to, instead of putting it into this machine that reads out kind of the fluorescence based off of how much virus there is, this is like you put it in a machine, you take out what comes out of the machine, you run it on a gel, and if there's a band on that gel of the right size, that would denote a positive. Now, if you have a really good and some of the more expensive tests, they take that band and then sequence it. And so then they can say, not only is this snake positive, but it's positive for a virus that is, 98% similar to ball python nidovirus. So it's probably just a new strain of ball python nidovirus. So that's kind of your different levels of testing. You have your cheapest test, which can tell you if a very specific pathogen that you already know about is already in your colony. And so that would be used for, like, I would use that to reoccurringly test my colony if I knew that test could pick it up. Then you have kind of your mid-range standard PCR tests, which are just going to tell you, yep, we detected a band that would be consistent of the right size for nidovirus. And then you have your really high-end tests that say, not only was it positive, but we sequenced it and it, came back as carpet python nido or ball python nido or you name it, or something different. We've had 3 new nidoviruses submitted to us like in the past 2 weeks.
Oh my
gosh! And like very different nidoviruses. Yeah, if it has a lung, it has a virus. We just haven't found it yet.
So to put that into perspective for people, how many different kinds of nidoviruses have you guys found? Roughly.
Oh gosh, I have no idea.
More than 20?
At the species level, more than 20 for sure. Probably closer to 30. And then within strains within that, probably there's got to be 100.
Okay, so it's a pretty widespread thing, is what you're saying.
Yes, it's a pretty widespread thing. And the only reason that we can detect all of those different types of viruses off of one test is sheer dumb luck. Like the primers that detect that virus, they got it right the first time.
Yeah,
the primers that we use just happen to be really, really good diagnostic primers at picking out a huge variety of nidoviruses. But those same primers, you could not do in a qPCR test. You just couldn't. They're totally different types. The way they operate is different.
So you might miss it or something like that.
Well, really, the thing is, these primers pick up a lot of different nidoviruses, but they also pick up a lot of other things. Now, you can filter out the stuff that isn't a nidovirus based off of size. So that's why you run it on a gel, and different bands will come out. The different bands equate to different size chunks of DNA. So a positive nidovirus test probably actually has about 5 bands on it, if not more. But only one of those would denote that it's positive for a nidovirus because it's at the right band length. Whereas you couldn't do that with a qPCR because it really doesn't make that distinction in the same way that this is a fragment was of the right size. It's really just what it was able to replicate. So, like, yes, you would get fluorescence from the nidoviruses, but you'd also get fluorescence from all the stuff that isn't a nidovirus that would really kind of throw off the test. So that's why you really need a specific set of primers that's only going to find that virus and nothing else. But what you're trading off for is that you can't find as many different types of viruses. Whereas the other type of test, your trade-off is that you can find a lot more different types of viruses, but you also find a bunch of other stuff too that you have to filter out based off size. So it's all these tests are. I wouldn't say they're just as much an art as a science because they're definitely squarely within the realm of science, but they're not as it's not just a magical thing where you take a swab and you put it in a liquid and it turns blue if it's positive or red if it's negative or something like that. It's
right.
There's a process and all of those processes have different trade-offs. So that's why testing can be kind of tricky because you have to make that trade-off of like, well, what is it that I am really looking for, and do I want a cheaper test so I can do more of them, or do I want a more expensive test so I know what I'm looking for? And so that's why it's also complicated. It's like, yes, you can get a $25 nidovirus test, but is that really telling you what you need to know? Probably not. So it's that's what's frustrating about it, is that there's a lot of different viruses. So it's not even just that we're talking about one thing. We're talking about a group of many things.
Right.
And so, the easiest thing you can do is to prevent it from coming in your colony in the first place. And that's just based off of quarantine and diet tests within that quarantine period. And then if you find that there is a virus within your colony, then you're really kind of looking through a practical lens of virus in my colony. Is it causing disease? Under what circumstance should I be scared of it? Maybe I need to do that snake last in the room. Maybe I need to make sure that I pay special attention to not cross-contaminate from this snake. Like, I have at this point in my colony virus-positive snakes.
Let's say that one more time. You were breaking up a little bit. Say that once more.
Yeah, I have four nidovirus positive snakes in my colony. I have 3 Canadian Samoas and 1 reticulated python. Both are adults that I've had for years that have never broken with a rest or anything like that. So that this indicates to me that this is probably one of those viruses that are just out there and probably not doing anything. But in the context of my colony, I have to make sure that I feed those guys last, that I'm making sure I don't cross-contaminate, and that I'm paying special attention that that virus isn't being circulated into a different species where it might be able to cause some damage. But at that point, it's like, well, I've had these snakes for 10 years and nothing else in the colony tested positive for these same viruses, so I'm just gonna let it be.
Yeah. And so for those people who think they hear nidovirus and they think death immediate, some of those snakes that have tested positive for you have been living for years and years and years, and they're adult animals, and they're not, experiencing symptoms, and they're not dead. So for those people who are thinking that nidovirus immediately means, death, and like when people hear inclusion body disease, IBD, they think immediate like death and destruction, and it's not that simple when you break it down.
Yeah, and really what it comes down to is that we just haven't done industry level testing for any of the things you're talking about. Like when you're talking about IBD or arenavirus, I don't think anyone's testing for arenavirus because I don't think they would be happy to find the answers they find.
Yeah, yeah.
And I feel like it's probably going to be pretty similar with nidovirus, where the prevalence is probably really, I don't even have to say probably, the prevalence is a lot higher than people think it is. but the industry is still here. It's not, they haven't all succumbed to disease yet.
Right.
So, and that's what makes it tricky, because which viruses matter in which context is not straightforward. So is Nidovirus instant death? No. But I also want to walk the fine line of making sure I'm not telling people that they don't have to worry about it, because you most certainly do.
Yeah.
Right.
Yeah, I can talk about my own colony, and the reason that again, this is no planning on my part to get into my current PhD position studying these viruses. I lucked, I guess you could say lucked into this position because I had a Nidovirus outbreak into my colony.
Mm-hmm.
And happened to be down the street from the institution doing some of the forefront research on these viruses. But in my colony of 150 adult animals, I had, I think, 6 different species of Nidovirus, and then within that, 9 different strains.
Wow.
And that's in one colony of 150 adult animals of I don't even remember how many species. Not a ton. Like, almost every species had their own unique virus. Like, this isn't something that these are viruses that have their own unique origins. It wasn't just that I had one snake with a virus and then
it spread.
That exploded into my colony. It was, no, these are, the viruses are out there in very high numbers, but in some contexts, it does nothing. And in other contexts, like in my colony, when I moved buildings, that stress pressure caused an outbreak of disease where, these viruses probably cost me about $250,000 in animals and sales. So, I don't want to say don't worry about it, because there's plenty to be worried about. But I also don't want to say that virus instantly equals death. It's all context and virus and species dependent. And honestly, it's probably even colony specific.
Yeah.
I've heard colonies of green tree pythons especially where, in their own colony, there's no disease whatsoever until they transfer, and then all of a sudden they can take their colony out. There are, I've had animals that are like kind of typhoid Marys that, like, they test positive. They've never, ever, ever broken with a respiratory infection. But every time I throw another snake in that cage to breed it with, they're dead within the year.
Wow.
That's crazy.
Yeah, that's insane.
Yeah.
Oh my gosh. Yeah.
Yeah.
Nidovirus, still so much to learn. And really it's really just a matter on the keepers, both novice and advanced, to just really do your due diligence when you're working in your collection, working to cut down any aspect of cross-contamination that you can, between, like, using hand sanitizers and like I know at Nerd we do a lot of the bleach sink stuff to disinfect, and then just making sure, like, you're disinfecting your surfaces that you're cleaning on, that you've got your tools, all that, to really just cut down that possibility.
Exactly. Yep. Yeah. And there's, a lot of it is kind of what we're doing now with the whole, social thing. You're trying to break the number of opportunities a virus has to spread. So how I manage it within my colony is it would not make sense for me, both financially or honestly logistically, to swap out gloves between every snake, or to do hand sanitizer between every snake. So, what it comes down to is I'm saying, okay, I have each of these racks as a unit, and so I swap gloves every time I transfer between racks. And then within each rack, I have my breeding group set up, so I'm doing hand sanitizer between each breeding group. And so it's all about just saying, okay, well, if I did have a virus-positive animal in that group, it's going to maybe have the opportunity to spread for, 3 or 4 other snakes in its breeding group. And instead, it's a little more contained than if I had just been like, okay, well, on to the next snake or whatever.
Right.
So it's just, and I'm at the end of the day, I'm a realist. If you wanted to ask a veterinarian about how you keep a perfectly healthy colony, it would be, swap out gloves between each snake. But let's be real, that's not feasible. So it's really just about, practical containment in terms of how do you keep something from, if you do have a problem, keep it from putting you out of the business, like it like it functionally did for me for at least a couple of years.
Yeah.
Mm-hmm.
Man.
That is intense. Definitely gives you a lot to think about, though. Not just for people who have got one or two snakes, but a lot of people in the reptile community don't just have just one species. They keep a variety of different things. They don't just keep one of a particular species. They keep a variety of different things, and it's definitely something for people to think about, especially going forward with who they're buying from, what they're buying, what their quarantine practices look like, what they're cleaning, and all that sort of stuff. All that plays into how this could progress going forward in the future.
Yeah, and I mean, it doesn't, virus or disease in and of itself doesn't instantly mean you did something wrong.
Yes.
Right.
That is the other thing that the industry has a really bad tendency to do is well, he sells diseased animals, so he's bad.
Yeah.
Right. Right.
And that's not the case. We know these viruses are in wild populations as well. It's not.
People didn't make it.
It should be, it should be not surprising that animals coming out of the wild, even if it's generations removed, still carry their microbiome with them.
Right.
And
right.
Viruses are part of that.
Exactly.
Man. That's some crazy shit.
Yeah. Every time I think about it, I'm like, oh my goodness, this is just, there's so much to it, and the more that you learn about it, the more that it kind of gets deeper and deeper and deeper. And so that's why I've been telling people, if they have questions about it, to go check out the recording of that talk that they did at Southeast Carpet Fest, that Steve and a couple other people who are doing research on the Nidovirus, to go take a look at the recording of that and learn a little bit, because there is still, they're still learning things, so we're still definitely learning, and there's this huge information gap that people just need to get on as soon as they can.
Yeah, for sure. And I would like to think, hopefully at some point, and maybe this is just me wishful thinking, that we'll be able to get to a point where, especially a lot of the breeders who do the reptile shows and stuff like that, all have the common, there becomes this common knowledge of what Nidovirus is.
Yes.
And how to answer the questions about it.
Yep.
Because I've heard from some people, they buy a snake and the breeder asks them, oh, well, are you buying from other people? You got to be careful because there's this deadly virus that's killing, whatever. And that fear installation is no good.
Yes.
So to be able to get to a point where everybody has at least some kind of common understanding of what is actually going on with this, I think that would maybe allow some of the chaos around it to simmer down a little bit, so we're not all
one would hope.
Yeah, exactly. I know it's maybe a little wishful thinking.
That seems a little optimistic based off of my experience in herpetoculture, but
I know.
We shall see.
I know. I'm
He's
trying.
A dreamer.
Yeah.
I'm trying. Damn it. I'm just trying.
Really,
we can't all go do pangolin research, damn it, Steve.
Well, that was the thing too. Like my experience with these viruses just opened up this and just honestly uncomfortable level of questioning and analysis at the world around us. So the world that we live in, where someone such as myself, along with all of the pathogens in the dirt on my shoes, can jump on a plane and go from Florida to Australia in 24 hours, that's a world that's existed within a human lifetime. Yeah. Before that, it was, you take a ship and you're there weeks from now where whatever's on your shoes is probably dead or tracked off in a different,
alive.
Right.
Yeah, exactly. Yeah. So the world that we live in, where there's pathogen spread, and again, this is a reality that I think the world as a whole is thrust into understanding in a very short time frame. The world that we live in is one that could be, scary in terms of disease spread. And so, when you look at chytrid and you think about where it's been, where have I been and where can that go in such a short time frame? And so I feel like the more we're starting to look, the more things we're going to find as it relates to this. And you know, that's a hard position to be in, is figuring out when they matter and when they don't, and what could happen to make them matter. Now, hopefully with the research that we're pushing out, it'll, you know, kind of at least have a framework for people to go to understand their questions. But I almost feel like there needs to be some intermediate, because, you know, some of the scientific writing can be honestly even jargony for my taste. It's something I've definitely had to get used to. So, you know, making sure that the information is not only in existence, but also, you know,
digestible
available
to everybody.
For people to fully exactly.
Yeah, man. Well, hopefully so. All right, so we're about at that time point where we got to wrap it up.
It's about that time.
So before we go, the one question we have to ask is, in any point or any part of the industry, be it something in your own collection or something that you have seen happening in the industry, what's out there that's got you excited? Be it like a morph project, a species project.
I mean, he did just get some scrub pythons.
He did get scrub
Yep,
pythons.
yeah.
I swear to God, if we have two scrub things back to back,
Yes!
we'll be really upset.
So so I'll do a quick aside real quick on the scrub pythons, because that turned into, basically I was able to get a pair really quick, because it looks like Florida laws are going to change where they're going to become really illegal really quick, where I luckily had the permits to have them up till this point. So those might be one of the last pairs of scrub pythons to make it into the state ever.
Wow.
Which honestly, if the laws change the way they're going to change, I'll never be able to breed them. So I have this pair of scrub pythons that I can look at in perpetuity, and that's it. But anyway, all that aside, what gets me excited about the industry? You know, lately I've been really on the species kick, and what started with that was the locality ball pythons. And like, if ever there was a weird thing to build a collection off of, it's weird that it started with objectively $25
ball pythons
in one context ball pythons. So I've been really excited in transferring over my colony to be a collection of rare, specifically python species. So I've been trying to get a member of each clade of python. And so what excites me is the people working with the weird stuff. And it's the people working with the weird stuff, that the stuff that was or is $25 now, imported for super cheap, the people who care enough to keep those going in a captive context, that excites me. So for me, it's my West African Samboas. I have a huge colony of captive bred West African Samboas that every offspring I produce, I could not give away. Like, they're imported for so cheap that, you know, no one cares, but they're just a stroke of a pen away from being totally disbarred from the industry. And you know, there's a lot of species out there.
Yeah.
And it's not even that they're unique. It's just that no one cares, and eventually that's going to be taken away from us.
Yeah.
It's not, maybe
That's
not today, maybe not tomorrow, but there will be a time when West Africa does not export any West African Samboa. And when that time comes, I'm glad I have a captive bred colony. So it's the people working with the scrub pythons, the people working with, you know, Timors, or any of the Indonesian ground or tree boa type stuff, the stuff that the Candoya that are imported for dirt cheap that no one cares about currently. I am really, I'm really glad that there are people out there that at least take the time to, hopefully at least there are people out there who take the time to try and captive breed them and establish them in captivity, because within my time in herpetoculture, which has been not that long, I guess, you know, a little over a decade, I've seen species come and go. I mean, there was a time when I walked past a Moluccan scrub python on a table for $200.
Oof.
Now I'm kicking myself.
That time doesn't exist anymore, and the clock is counting down on all of these species, every single one.
Yeah. But
we don't know when that timer ends. So anyone who's taken the due diligence to breed or keep a colony going of anything that no one cares about now, that I'm grateful for. Like, I give you a specific example. What is it? Joe Bass, the girl working with all the different samboas that has all the different types of samboas, like all the Arabians and stuff like that.
Yeah.
Like, now that Arabian samboas are a meme, that she's like the only person in the country I would know to ask about them.
Yep.
Yeah, that's very true.
I'm
That
with
is
it.
very true.
So, Steve, if people wanted to learn more or get in contact with you, how would they do that?
I don't know. Just go email yourself. I'm probably not going to reply.
Damn.
That's pretty close to my
He's
answer.
too cool now. He's in a hazmat suit in the lab telling you to fuck off.
Yeah, he's doing his PhD. He's gonna be Dr. Tillis. You know, he's got too much stuff going on.
I do. I would say probably Instagram's the best. I have an Instagram at Reptilis. That probably is going to be the best place to follow me. I don't post much on there. Again, I don't have a desire to talk with or deal with customers anymore. So
don't hit him up. Just don't hit him up.
Just
follow him.
Enjoyment,
Appreciate
I guess,
the pictures.
But yeah.
I'm with it. I'm with it. Cool.
All right.
So that's going to be a wrap on episode 3.
That wraps it up, dude. Thank you so much
very much for
hanging out
Steve.
with us.
Absolutely. Thanks for having me.